Welcome to this live broadcast where Scandion Oncology will present their positive top-line Phase IIa data from the Corist Part 3 trial. After the presentation there will be a Q&A and we will also raise questions from the live chat, so please go ahead and ask them there. To start this off, I'm happy to introduce Scandion Oncology CEO, Mr. Francois Martelet. Please welcome.
Thank you very much, and welcome to our Research and Development Day, so-called Investor Day, to all of you who are online right now, and also to those of you who are attending physically this meeting. Well, this is the traditional disclaimer that we don't need to go through. Well, and I will start, first of all, to give you a company overview, and then we will go into a lot of details from a scientific perspective, clinical perspective, and other matters as well. So as you all know, Scandion Oncology is a biotechnology company listed here on the First North Stockholm Stock Exchange, and we are based in Copenhagen. We are a small company. We have been recently right-sizing the company in order to preserve our cash and our level of capabilities as well, and we are exclusively focused on oncology. Yes, so this is a slide about our shareholder information. This is a busy slide and we don't need to go through that into details. But despite the fact that we are a company based in Copenhagen, the vast majority of our shareholders, our retail shareholders, are based in Sweden. That's the reason why we wanted to do this R&D event here in Stockholm. We have approximately 8,000 shareholders, which corresponds to approximately also 40 million shares. And the vast majority of our shareholder base consists of retail shareholders. Right, so on the right-hand side of this slide, this is a management team. You can see myself, and I would like to introduce the other players in our management team. So we have Johnny Stilu, who is a very experienced CFO. He's here in the assistance. You will not hear from him, but of course, Big Brother is watching me and the others as well. We have Lars, Dr. Lars Damstrup, our Chief Medical Officer, who will go through all the clinical data, not only the Corist 2, but also Corist 3. And we have also here Dr. Stenvang, our Chief Scientific Officer, who is also a co-founder of Scandion Oncology, who will go through the science. And then you have an opportunity to ask him as many questions as you wish. You see also our board at the bottom of the slide, which consists also of fairly senior level executives in our industry. Right, so this is an important slide. This is our advisory board, our scientific advisory board. And as you all know, a good technology is always endorsed by key opinion leaders. And we are fortunate enough to belong to this category. And I would like to have a special mention for Dr. Josep Tabernero, with whom we are in very close contacts and with whom we are asking for advice, and it's always a back and forth relationship, very useful for us. Obviously, we are following a lot his guidance. And by the way, we recently held an advisory board meeting, and then we have also added other names from Europe, but in order to review our clinical data, in order to get advice on the next steps from a clinical trial perspective. And this is very important to have this level of guidance for us. Right, so this is our pipeline. We have a very innovative technology, groundbreaking, in a way that very, very few companies are in this field. We are in drug resistance. That means we are working on ways to decrease resistance to drugs used in cancer. We are working in the efflux pump inhibitors in colorectal cancer and in pancreatic cancer. Jan will go through this into details in our mechanism of action, which is rather unique in our industry. We have two compounds, as you see from the slide, SCO-101, SCO-201, We have, for a number of reasons, prioritized SCO-101. It doesn't mean that SCO-201 is a bad compound. It means that mainly for financial constraints, we have prioritized the clinical development of SCO-101. And we are working, as I said earlier, in 2 indications, colorectal cancer and in pancreatic cancer from a clinical perspective. We have also worked a little bit in gastric cancer. We have gained positive data in preclinical data in gastric cancer. Right, so we are a small company, of course, as you all know, but we still do have a very experienced management team, and we are also working externally with world-class suppliers. A lot has been done over the past 2 years, and if we look at, I joined the company at the beginning of 2023, so about a year ago, and we have done a lot. We have certainly, released our data, Corist 2, Corist 3, and we have a strong IP. I will come back to that. We have manufacturing capabilities in place as well. We've been seeking regulatory advice on our clinical program, and all together, we believe we are in a very strong position to move forward. So having said that, now I will turn it to Jan, our Chief Scientific Officer, to go through cancer drug resistance. Jan, please.
Thank you, Francois. Hello, welcome to all of you. Just a few slides on the background for cancer drug resistance and a lot about the science, and you will have opportunities to ask questions after the talk, obviously. So cancer drug resistance is a major problem. There's a large unmet medical need. As you can see, there's on the first bullet point, we regard that there's 2 different types of resistance. One is acquired, where the patient has an initial response to the drug, and then later something molecular changes happens in the cancer cells and they become resistant. That's one thing. The other one is that the patient doesn't respond to the drugs in the first place, so we call that intrinsic resistance. One of the molecular changes that may happen in cancer cells, there's more on the next slide, is that the chemotherapy is actually expelled or pumped out of the cancer cells, thereby, of course, not giving the chemotherapy the opportunity to work inside the cancer cells where it's supposed to work. Here are some general mechanisms of drug resistance. Drug efflux pumps are not the only mechanism. I guess everybody in the field is familiar with that. Here you can see that bullet point number 1 is exactly what we are addressing. It's the efflux of drugs. I'll come back to more details on that. However, it's also important to say that there could be other options, such as decreased uptake of the chemotherapy or changes within the cells. The target may change, the cancer drug may be metabolized in a different way, and so on. But the drug efflux pumps has consistently over the years been described as a major and important part of cancer drug resistance. And one thing that I would like to pay attention to is the sentence on the bottom of the slide, that 90% of cancer death has been ascribed due to resistance against the current treatment. So there's a huge unmet medical need in cancer drug resistance. This is what we're trying to do something about. SCO-101 is, of course, as you know, our lead molecule. It has a dual mechanism of action that I will try to go through now. There's 2 main things here. On the top line, you see that we have some effect on the tumor that addresses the efflux pumps. If we can inhibit the efflux pumps, we will have more chemotherapy inside the cells. I'll come back to these points. The other one is more than a plasma effect, where we have the potential to increase the level of chemotherapy in the plasma of the patients. So the first one, the efflux pump, the point of pumping out the chemotherapy of the cells is this targeting of ABCG2. So in the initial setting where the chemotherapy works the way it's supposed to do, you add the chemotherapy to the cells, or in the patient setting you give the chemotherapy to the patients. It accumulates inside the cancer cells and it will hopefully kill the cancer cells. However, On the bottom line, you see this arrow representing the ABCD2 effluxing the chemotherapy, gets out of the cells, the chemotherapy doesn't work anymore, you have resistance. What we think we can do with SCO-201 is to put a plug in this pump. As you can see on the bottom line on the right hand side, SCO-101 blocks this ABCD2 and the chemotherapy is restored inside the cancer cells, meaning that we can kill the cancer cells again. That should be hopefully easy to understand. We have, of course, done a lot of data analysis on this in the preclinical space. And you see on the right-hand side, we have made some enzyme assays to inhibit 3 major efflux pumps named ABCD2, as I just said, but also ABCB1 and ABCC2. What this curve represents is the enzyme activity, and what you can hopefully appreciate is that the left-hand side is ABCD2. It has an inhibition where 50% is inhibited, called the IC50 value, which is low, 0.15 micromolar. That means it's much more potently inhibited than the 2 other ones, the yellow and the blue one to the far right. So that means that we have both a potent but also a specific inhibition of ABCD2. On the right-hand side, we have made an assay where we have looked for substances to pass a plasma membrane. On the upper side, we have a substrate that is transported by ABCD2. The high column to the left is what happens when SCO-101 is not in the system. It represents a large flux through this membrane. And then you can see when we add SCO-101 to the right on the top panel, it totally blocks this flux. So SCO-101 blocks the flux of this estrone 3 sulfate through the membrane. So we also did that for ABCB1. It's the lower part of the slide. And on the left-hand side, you can see this substrate for ABCB1 is transported through the membrane. And when we add SCO-101, nothing happens. So again, it supports the notion over here. We have a specific inhibition of ABCD2. So we have also looked what does that mean in terms of cancer cell. Can we actually show that chemotherapy actually accumulates inside the cancer cells? On the left-hand side, it's not chemotherapy, it's a dye, but this dye is a substrate for ABCD2. So if ABCD2 is present, it will be pumped out of the cell. On the top line, we have a cancer cell line, a colon cancer cell line with low to no ABCD2. And you can appreciate when you add to the blue cells, it's the fluorescence microscopy. When you add this dye, the cell is accumulated inside the cells. You can see it by the blue dye. We have then a chemotherapy resistant cell line, which is the middle panel. This has a high level of ABCD2, and when you add this fluorescent dye, you can see it's all black, meaning that this dye is pumped out of the cells. And most importantly, on the bottom side, you have what happens when you add SCO-101. You can see that the cells turn blue again, even though it's this resistant cell line, and you restore the accumulation of the dye. So that was just the dye. So we also tested on the right-hand side if that also applies to real chemotherapy. So we took the chemotherapy and we radiolabeled it. And what you can see over here is that on the y-axis is the amount of chemotherapy inside the cell. And on the x-axis, it's increasing concentrations of SCO-101. So when you increase the amount of SCO-101, you get more and more chemotherapy to accumulate inside the cancer cells, exactly what we want. So we also tested if this has any impact, any effect on the cancer cells, or it's just something that we have observed. So on the left-hand side here, you see it's a little bit busy slide, so I'll walk you briefly through it. On the y-axis, we have the viability, meaning how many cells is alive. It's an experiment done in a petri dish, so we can easily measure the number of viable cells. And you can see when we start, it's all 100%. So all cells are alive from the beginning. On the x-axis, we increase the amount of chemotherapy, going from low to high. The red solid line represents a resistant cell line. The green solid line represents a sensitive cell line. And you can see that these two cell lines have very different levels of ABCD2, the efflux pump. The red one, the resistant one, has a high level. The green one, the sensitive one, has a low level. And you can clearly see that you need much more chemotherapy to kill the resistant cells. So that's fine. The system works. What then happens when you add SCO-101 is when you look at the red dotted line, you restore the sensitivity, you get the sensitivity back to the level as for the sensitive cell line. So this is exactly what we hope for. We can resensitize to chemotherapy by adding SCO-101. On the right-hand side, there's two traditional, preclinical experiments. The first one is to look at clones, the clonogenic potential. And what this shows is that if you add SCO together with SN38, the yellow bar, you get a much stronger suppression of the clonogenic effect, meaning suppression of the cancer clones to grow up. So that is also something that is very important because we want, in a patient, of course, we want to kill cancer clones. On the right-hand side, far right, we have tried to test short exposure where we have the chemotherapy and the SCO present at the same time and look for longer effects. The dark bars is 3 days, the lighter bars are 6 days, and as you can appreciate to the far right, when you add chemotherapy and SCO-101 for only 24 hours and look after 6 days, you have a very strong effect. We hope that would be the same in the patients, where we have some windows where we have strong exposure to both chemotherapy and SCO-101 on the cancer cells, and that these effects will also translate into longer term effect. Okay, so that was the ABCG2, a little bit of data on that. I hope I convinced you that we do inhibit ABCG2. So the other target we have is UGT1A1. I'll just show you how the system works, and then I'll show you how SCO-101 may play into that system, and in the end, how UGT and ABCG2 will play together. Here's the system. The patient receives irinotecan. Irinotecan is a prodrug. It's converted into this active chemotherapy named SN38. This SN38 is converted by this UGT1A1 liver enzyme into a glucuronidated version, just G for short, which is inactive and secreted out of the cells. So this is the active part. What happens then when you add SCO-101? You block this enzyme, as I showed you before, and we have an accumulation of active chemotherapy and, of course, lower levels of the inactive chemotherapy. So that's the system when we block UGT1A1, more active chemotherapy. Here we have made two different enzyme assays with UGT1A1. Beta estradiol is just a substrate you use in the experiments, which is a standard assay. But of course, we also use the real chemotherapy, and you can see that the conversion of the real chemotherapy is decreased when you add SCO-101. The IC50 value, this value where you have 50% inhibition, is very low, 0.1 micromolar over here. And we also tested other UGTs, so you can see it's quite specific. So we have also here a specific and potent inhibition of UGT1A1. That actually correlates quite well with all the previous data we have, both in normal healthy volunteers and also in our own trials. Lars has a slide on that later. I just want to say that the gray bar is what we have calculated when the patients get normal irinotecan treatment, how much SN38 is in the plasma. And when we use SCO-101 and 50% FOLFIRI, we get a much larger exposure of SN38, the active chemotherapy, in the plasma. So we expect the cancer cells to react more, obviously. Now it gets to the point where we will try to combine inhibition of ABCD2 and UGT1A1 in a cartoon, trying to show you what we think this can do. Here's the normal system. We have no SCO-101. We have low levels of ABCD2, sorry. So we have the conversion of SN38 by UGT, and we get some accumulation inside the cancer cells down here. This is what you should focus on. So we will have an effect there, and that will be an effect of some magnitude. If we then take the situation, same situation, but we have this resistance, the ABCD2 in the cancer cells, this will efflux the chemotherapy and we'll have very low levels of chemotherapy inside the cells, and of course we expect that the effect of the chemotherapy will be low. What happens then if we add SCO-101? Now we take this situation and we add SCO-101. You can see we will have more active chemotherapy. That will of course results in more chemotherapy inside the cells, even more than in the starting point over here. And then the last point with the patients who have increased ABCD2, if we add SCO-101 into that system, this is what we expect would happening. We have the same blocking of UGT1A1, more active chemotherapy, more accumulation, and we will also block the ABCD2, leading to even further accumulation of chemotherapy inside the cells. So this is how we see the effect of this dual mechanism of inhibition. Okay, enough for SCO-101 and the dual mechanism of action. We also have SCO-201, as Francois briefly alluded to, is something that we would like to explore further if we have the opportunity. So the cancer potential is quite clear for us, and this has also been published. SCO-201 is a very strong inhibitor of ABCD2. We have also tested in vitro that it inhibits UGT1A1, and it does. So there's a lot of things that we would like to do there, and it has been tested. What happened there? Okay. Okay, we're back. Thank you. We have also tested in human hepatocytes, and it seems to be very non-toxic up to high levels. So we think this has a good future. Antiviral potential is something that we have not talked much about. It has been published in the literature, and there's some types of viruses where we can see that SCO-201 actually inhibits the replication of these viruses. So that could potentially lead to exploration of other indications. We know that ABCD2 has a role in HIV, both in terms of expression on some of the cells that HIV infects, but also in a role for the efflux of these drugs that are used in HIV. And as you can see to the right side, there's a massive need. There's almost 30 million people receiving treatment for HIV worldwide, and 50 to 90% of these patients are falling short due to resistance. So there's a massive potential here that we would like to explore further. So thank you very much, and with that, over to Lars.
Thank you, Jan. So my name is Lars Damstrup. I'll walk through the clinical data for the SCO-101. So this here is as you know, Corist is our most advanced program. We have conducted Corist 1, 2, and 3, and I'll touch base with these 3 segments. In all cases, we have had patients with metastatic colorectal cancer. We have evaluated different schedules, etc., which I'll go into more details on the next slide. So Corist 1 was the initial study, which was an MTD, which means that we were looking for the toxicity or the safety profile of SCO-101 in combination with standard of care FOLFIRI. And SCO-101 in that part of the trial was given on a 6-day schedule, so day 1 to 6, and FOLFIRI was given on day 5 to 7. We used a dose escalation from starting with irinotecan and 5-FU at 80%, and then that was too toxic, so we went down to 60 and then to 50 milligram. And we established the maximum tolerated dose, and the recommended phase 2 dose was in part moved forward into Corist part 2. And we treated 18 patients in that part of the trial. Then in Corist part 2, we had a segment of just KRAS wild type patients with metastatic colorectal cancer, and we treated 25 patients. We used 50% of irinotecan and 5-FU, and 150 milligram of SCO-101. And that was given also the same schedule as in Corist Part 1. And the main objective for that part of the trial was objective response. As we communicated in, I think it was September 2022, we did not meet our primary endpoint, which was a response rate of a certain percentage. However, as you all know, we in November last year communicated the top-line result with quite an impressive overall survival of 10.4 months. I'll come back to that a bit later. Now, Corist Part 3 was also a study where we wanted to test two different schedules. You could say we were trying to optimize the treatment schedule for SCO-101. So we had two different schedules, either the 6-day schedule, as seen on the left part of this panel on Corist Part 3, and then a 4-day schedule, which is shown on the right-hand side. And we used different dosing from 150 to 250 milligram. We established the maximal tolerated dose for the 6-day schedule with a 150 milligram of SCO in combination with 50% irinotecan and a full dose of 5-FU and sulfuric acid. Now, we have also then we used the 4-day schedule, and we did not see any toxicity, or so we found it extremely safe to give the 4-day schedule with up to 250 milligram of SCO-101. We established that we would declare this as our maximal administrative dose. I'll come back to that towards the end of my presentation. Now, as Jan also alluded to, and he showed this, and I'll not go into all the details because Jan walked us through the dual mechanisms of action. But on this slide is very, very impressive, looking at the blue line, which is a 50% dose of irinotecan, and full dose and the gray is a full dose. So we can show here that we have a higher exposure over a longer period of time. So from a cancer cell's perspective, this is their worst nightmare, leading to an increase or potential increase of tumor cell death. Now, the key findings from Corist Part 2 was, as I said, we have an impressive overall survival in the 25 patients included. And but in historical data, we have seen that the overall survival in the same patient population is in the ballpark of 5 to 7 months. So we are certainly increasing the overall survival. We saw a progression-free survival of 2 months, which is completely in the ballpark what is seen in other studies in the same patient population. We also saw what is known as a clinical benefit rate, which means that patients who have a tumor regression or better, we saw that after 16 weeks, that was 20%, but after just 8 weeks, it was 42%. And again, in historical data, the clinical benefit rate after 6 weeks has been reported in the ballpark of 10 to 16%. And as I said, we saw 20% tumor reduction. That means that 5 of the 25 patients had some tumor reduction, but not to the limit or to the extent that it was very deep. We also throughout Corist Part 1, but also now in Corist Part 2, we could identify a subset of patients with a bilirubin that was transiently increased. So it means after 7 days, it was increased, and after 12 days, it was decreased. I'll come back to this in a moment. So this is a standard Kaplan-Meier plot. On the y-axis is the percentage of patients without an event, and on the x-axis is the time frame, and this day, excuse me, and this is in days. The solid line is progression-free survival, and the dotted line is overall survival. And if you can imagine a line going from 50% crossing those two curves, this is where the median, so the median progression-free survival and the median overall survival are read from these curves. As I said, we had a subset of patients where bilirubin was increased and then rapidly decreased, and that was and that's a solid line, and there was a dotted line is the ones that did not have this. They rose, but they stayed high, and so we believe that this bilirubin evaluation could serve as a potential biomarker down the road, which is a very important parameter. So in the patient that have a transient increase in bilirubin, they had a median overall survival of 13.4 months. And this is the solid line, whereas the dotted line, for those who had a persistent increase, it was only 8 months. But still, even those had the same overall survival as compared to the historical data, which is between 5 and 7 months. Now, part 3. As I said, part 3 was initiated to further evaluate and elaborate and refine dosing and scheduling of SCO-101. We had 2 dosing regimens. On the left-hand side is dosing regimen 1, where we started with 150 milligram. We saw absolutely good safety and tolerability, and increased the dose to 200 milligram, and saw 2 patients out of the 4 where there was a DLT, which is the limits that we put in the protocol to say if a patient has this and this toxicity, it becomes something that we need to be aware of. And then if we had 2 or more, that means we could not further increase the dose. So therefore we went back to the 150 milligram. So we have, and there we did not see any safety and tolerability issues. So therefore, in this dosing schedule, we established the maximum tolerated dose. And now everything goes black.
Not in the broadcast.
Okay. Yeah, but I can't see anything.
Oh, there too.
Yeah.
Okay.
It's got back. And then we went into dosing regimen number 2, which was giving over 4 days only. And we started with 200 milligram and then went up to 250 milligram. And we did not see any safety and tolerability issues. So we established, we concluded that the 250 milligrams giving 4 days in combination with 50% irinotecan and full dose of 5-FU was a maximal administrative dose that we have given. However, I'll come back to that right at the end. Now, this is a progression-free survival of the Corist Part 3. It's still, the follow-up is still ongoing, so we don't have any overall survival data at this point in time. And what's interesting is that we had a median progression-free survival of 4.6 months, which is even better than we saw in Corist Part 2, and certainly also better than the historical controls. There are 3 still patients under treatment, and we also saw that the clinical benefit rate after 6 weeks was about 76%, and I think I'll summarize this on the next slide. Yes. So the key messages or key findings from Corist Part 3 is that we see a promising median progression-free survival of 4.6 months. We have an exciting clinical benefit rate of 76%. And we have in this part of the trial have had 1 patient with a more than 30% reduction. So this means the patient has what's known as a partial remission. And then there's 1 patient with a tumor reduction close to, but not quite 30%. But in 15 patients, we saw tumor reductions less than 30%. And again, we have seen some of the same trends with respect to the bilirubin. So we are working on this to further elaborate these data. So the lessons learned and the next steps for Corist. First of all, as I said, we have impressive median overall survival. We have tested many different dosing and schedules. We have exciting clinical benefit rate. We have successful tumor reduction. So the question becomes, of course, what is our next steps? as I said, we have the maximal administrative dose in the Corist Part 3. The next logical step for us is really to have what's known as a proof of concept, which means that we will do a randomized phase 2 trial with, I'll come back to the setup for that in a moment. However, for us to, shall we say, maximize the efficacy of SCO-101, we as an enabling study can foresee that we will increase the dose of irinotecan with this dose of SCO-101 that's been used in Corist Part 3, Schedule 2, as an enabling study for the randomized phase 2 trial, thereby, shall we say, replacing what's the Corist Part 4, which I'm sure many of you have heard about, which I have not presented this for these reasons here. Now, the randomized phase 2 trial is a classic setup with the patients that have metastatic colorectal cancer. They would be eligible for the standard of care FOLFIRI and will be randomized 2-to-1. So we'll have 100 patients that will be treated with our drug and FOLFIRI, and 50% that will be treated with the standard of care out there, which, as also Francois alluded to, we have had in December we had an advisory board meeting with Tabanero and 5 other European key opinion leaders, and they endorsed this concept and were very thrilled about the data that I have shown you today, but certainly they were very much in favor and would all like to participate in a trial like this. So, the control would be regorafenib. It's sometimes difficult to say this. And then the study would continue until either patient had progression, or until the patient did not want to stay on a trial anymore, or if the investigator believed that the patient did no longer had a benefit for the trial. So it's pretty standard. Keep on going as long as you can. And then the follow-up would continue, and the primary endpoint in this would be overall survival, which is a very hard endpoint. It's because which is accepted for regulatory purposes, both by FDA and EMA. Now, so much for Corist. Now PANTAX. I'll just got one slide on that. The PANTAX is a trial it's a 1B trial in patients with pancreatic cancer, and it's a phase 1B. So all patients, we included patients from Europe on a 28-day schedule with SCO-101 given again day 1 to 6. Gemcitabine was given weekly, and gemcitabine was given every 3 weeks. And now both gemcitabine and paclitaxel were given at 80%. The patient population, as you can see, pretty standard. The primary endpoints was to determine the safety and tolerability in combination with gemcitabine and paclitaxel, and to further evaluate the maximal tolerated dose, and therefore have a recommended phase 2 dose. And the secondary endpoints were, again, efficacy parameters such as objective response rate, clinical benefit rate, survival, etc. And then the interim findings were, first of all, we published some of those data from the safety and tolerability point of view. We published that at ESMO last year, and so later this year we hope to have the full data set, and we'll communicate this to you as soon as we have that. And with that, I'll leave it back to Francois.
Thank you, Lars, and thank you, Jan, for a deep explanation about from a scientific perspective as well as from a clinical perspective. I hope you're not overwhelmed by the data. And now that's the easier part of the presentation. So I thought it would be useful for you, to understand what are the thinking of our key opinion leaders around the world. And if you look at this slide, and again, this is coming from a blinded study. So we ask, a company X, to ask to key opinion leaders, what do they think about our compound, SCO-101? And we present the set of the data, and then they will tell you what they think about it. And if you look at this slide, they would say, well, the inhibition of both ACPG2 and UGT will certainly increase the concentration of drug metabolites. Great. And then also, the therapeutic options here are scarce. Great as well. We don't have any great treatment that can either prolong or reverse cancer growth. And I think this is a very important point mentioned here. UGT inhibitors would be very effective against pancreatic cancer compared to other malignancies. So this is the feedback, coming from our key opinion leaders that we actually don't know. Again, this is a blinded study regarding mechanism of action, regarding also the indication pancreatic and colorectal cancer. Now, on the data itself, look at what they say. PFS gives us a better look at whether that drug is working the way it is supposed to. Another one about the potential biomarker. Identifying a biomarker makes a lot of sense. There are certainly patients with UGT1A1 mutations that I would not give this to. And actually, we all know that without a biomarker, FDA will probably nowadays will not approve any oncology drugs. So we are actually, it's very important that for us that we continue to work on a biomarker, as Lars was mentioning. Right, and now a few words about, IP, because I think it's extremely important. We used to say in our business, no IP, no business. And here we are very fortunate to have a very strong IP protection for our main compound, for our lead compound, SCO-101. We have the combination treatment IP has been granted in most of the countries. And then we have 7 patent families for which the patent is pending. Kinase inhibition, PK optimization, bilirubin index, crystal forms and amorph and salt as well. So for this is pending, but we have already the combination treatment with chemotherapy granted, which is obviously very important, up to 2037, and some of them 2042, as you could see from this slide. Now, SCO-201, although we have not been able yet to develop this compound from a clinical perspective, we know from a preclinical as well, we have already started to make the steps moving forward from an IP standpoint in order to protect the compound and therefore the company for its future development. Commercially, what are the commercial opportunities? Colorectal cancer is a very deadly disease in oncology. This is the third major indication in oncology. You can see the incidence rates here in Europe and in the US as well. You could see also the 5-year survival rate, stage 4, colorectal cancer. This is, as Lars said, this is the indication in which we are in, 12%, you see. So there is obviously a major unmet medical need. And you see here you can see here where with the star, the red star, where we could potentially treat the patients with colorectal cancer late stage in combination with either FOLFOX or other drugs as well. And that's an important, obviously, we would like to move up, the design of the clinical trial down the road. But I think it's important for you to clearly understand that this is a big market. Right, and talking about big market, I won't go through that, but, we have a process in market research and forecasting, in order to calculate roughly the potential of our drug, and you could see here we are well beyond the $1 billion drug, and this is classical, the definition of a blockbuster drug, provided, of course, if we have approval of the drug in combination with chemotherapy down the road. Right, a few words about gastric cancer. Gastric cancer is even more serious, if I could say so, from a survival rate perspective compared to colorectal, because you can see here the 6% survival rate. Although we are also in early stage of development, only we have preclinical data, positive preclinical data, but clearly you could see also the ACBG2 expression as mentioned by Jan earlier. And this is the sixth most, let's say, most deadliest disease in oncology, and with obviously a significant market potential as well. Now, a few words about manufacturing. So we are working with world-class suppliers, both from a drug substance perspective, also from a drug product perspective. From a drug substance perspective, we have an excellent stability at room temperature, that's important. And from a manufacturing, drug product manufacturing perspective, we have been working with that vendor, and we are fully compliant from a phase IIa perspective. Obviously, if we want to move for the phase III, we will have to identify another supplier. And also, we have been seeking regulatory advice on that as well. Right, so in summary, if you look at last year, we've been able to close studies, analyze data, report data to the market, and report successfully data to the market. So 2023 has really been a year of turnaround of Scandion Oncology. If you look at what has been even released very recently, the Corist top-line data, looking further, so we have the PK study for Corist to be released on the road. We have, during the first part of this year, the Corist final data of Corist part 3, and the PANTAX phase Ib result, as our CMO mentioned. So there is a lot still to happen at Scandion. And having said that, I would like to also, before I open to the Q&A, and before I close down this presentation, I would like to thank all our shareholders, our retail shareholders, because we know that it takes some patience for you. It takes a lot of time to develop a drug, and there are so many steps in between, and we understand some of your frustrations from time to time. But we really appreciate your trust and confidence in our company, and stay tuned. It will be even more exciting in the near future. So having said so, I will turn it to you for the Q&A sections.
Thank you. Thank you so much, Francois, and thank you, Jan and Lars. And if I start with you, Francois, you have a long track record in big pharmacos and within biotechs as a CEO. Why did you join pretty small Scandion a year ago?
That's a very good question. I was really impressed and fascinated by this dual mechanism of action that almost no one has tried before. And this is super exciting. This is very innovative, and there is a rationale. We've been able to demonstrate preclinical data, clinical data. So obviously we need to go further into phase II, phase III, moving to close to regulatory approval. But, we are probably 2/3 of the way, but it's super exciting, And then, to have a compound that really increases the power of chemotherapy is absolutely needed, in our armamentarium of drug in oncology. So super exciting from a mechanism of action and clinical data as well.
You couldn't do anything but join them.
Yeah, absolutely.
So what is your opinion on why is it so difficult to thrive within biotech in the Nordics?
Yeah, biotech is, it's very risky. As you see, at each step, you have a gun in your head, saying, okay, will I get good data or not? So this is the risk adverse. The risk is one element. Two is that obviously in the Nordics, the vast majority of the shareholding base consists of retail shareholders, and there are very few institutions, so it doesn't really help to let's say to raise money with big numbers. So that's more difficult than in the US, for instance, where you have a different kind of institutions and shareholding base. But clearly, I think that retail shareholders do appreciate what we are doing, despite our, it is difficult, it is risky, difficult, it is long, it takes so much time to develop a drug, moving from one phase to another. And, again, I'm very thankful for that,
Mm-hmm. And it seems to me like you've done a turnaround within this year. Could you elaborate a bit on this?
Yeah, well, it's, we've been fairly quiet for the first part of the year, last year, let's say 2023, because, it takes some time, to reflect, to analyze the data, to look for ways to optimize the design of your protocol, of your trials, moving a 4-day schedule, 6-day schedule, etc., looking at the capabilities of the company itself, basically, we wanted to be, to have a small team of very senior executives like we have now, and then, basically outsource whatever we can in order to preserve cash, in-house cash, and capabilities as well, but without, without, let's say, losing too much of capabilities. So that was the kind of, it's a delicate balance, what you have to retain internally vis-a-vis what you outsource externally. But we've been pretty successful at that, and I have to say that we have a burn rate that is very consistent, and with the burn rate of a small biotech company, putting a lot of effort in our trial, putting a lot of money in trials, but this is the lifeblood of the company, basically.
Okay, and I'm really interested in your answer on this question. In last line, colorectal cancer patients, what is the ultimate parameter of success?
Yeah, and that's a question that I have addressed a bit earlier on recently. Last line patients, what counts ultimately is what? Is obviously overall survival. Of course, progression-free survival, the interval by which they are more or less in remission, they can live normally, is very important. But the overall survival is extremely important. It doesn't mean that the basic parameters, tumor shrinkage is not important. But if you're a last-line patient, what counts is really the time that will the treatment will give you to live longer.
Absolutely. Thank you. Shall we bring Lars Damstrup up on the stage again to hear some more?
Okay,
thank you. Thank you for the presentation earlier. In total, how many have been treated with SCO-101?
Over the years, more than 150 patients. Some of these have been healthy volunteers, and you can add up the numbers from our presentation to see how many cancer patients have been treated. But it's more than 150 in total.
Thanks. You've mentioned pancreas and colorectal cancer. Are there any other indications?
One thing that also Jan has mentioned and Francois also alluded to, I think that because of R&D can also being used extensively in gastric cancer, this is very obvious indication to explore as well.
And overall, with the experience with SCO-101, what is the next logical step?
I think I mentioned this, that going into the randomized phase 2 trial is our ultimate goal, with an enabling study to optimize the dosing schedule for, that we have established in Corist Part 3, the second schedule, so the 4-day schedule.
And in your experience with SCO-101, you mentioned that it is well tolerated.
Yes.
What toxicities have you seen?
First of all, you have to acknowledge the fact that our drug is given with the late line of therapy, where patients have been treated with a lot of chemotherapy. We are combining our drug with chemotherapy, which is by itself, by nature, is toxic. So in our protocols, we have established that we have seen some patients with neutropenia, which is manageable. And if you remember, on one of my slides, our overview of Corist 1, 2, and 3, in Corist Part 2, we mandated that patients should be given GCSF, which is a drug that can increase the bone marrow's production of white blood cells, minimizing the risk of neutropenia. We do not endorse, or we do not mandate this further on, because we have seen this neutropenia grade 3 or 4 in a very few patients. So giving a potential very risky drug, we're not going down that route again.
Okay.
So as I said, it's very safe and tolerable. We do not see any major toxicities apart from what we would expect from chemotherapy backbone.
Yeah, the mixture there. Yeah. And I also received some questions from Red Eye equity analyst Christian Binder, and he has a few questions. I think they are suitable for you to answer. Based on Corist Part 3 result, do you still believe Corist Part 4 is the best way to proceed with SCO-101's clinical development, or do the data motivate a change in your clinical strategy?
To reiterate, as I said also, we do not believe Corist 4 is a best way forward. I believe as an enabling study for the randomized phase 2 to optimize the schedule of SCO-101 in combination with a higher dose of irinotecan, this is our best bet.
And another word from Christian Binder. Corist Part 3 data look promising compared to the cited historical controls. Still, it is often hard to draw strong conclusions based on such comparisons. When do you estimate that you will be able to present randomized collateral data with SCO-101 in CRC?
First of all, in order for having a randomized trial, which I said we have had this evaluated also by an external statistician, we need 150 patients. And of course, in order to have this, we need money. But even if we had the money today, they will take at least 3 years plus. I cannot go into more details because it depends on how many patients, how many sites would be enrolling, how fast we can get patients, 150 patients in, and also how or when we see the events. Because as also Francois mentioned, and also I put on my slide, the primary endpoint for a randomized proof of concept in this patient population would be overall survival. And we saw from our earlier data that patients with at least with a bilirubin index that was transient, also it went up and then down, we saw a 13 point, I can't remember, it was 13.6 or 13.8 months of overall survival. So therefore you need to have patients, the last patient needs to be observed for at least a year or a year and a half before you have the conclusion of the overall survival data. So I cannot give you an estimate.
No, but good enough. Do you believe Corist Part 3 data could suffice to enter a partnership or licensing deal for continued SCO-101 development?
That's hard, and I'm not an expert in this, but I think that having written a protocol for the randomized phase 2 trial for a potential partner to also to look at could be a very good incentive. Okay. I don't know if my colleagues have further insight into this. I'm just a simple clinician.
Okay, and here's another one related to your job. What can you say about the 4 patients that were not evaluable in Part 3?
2 of those were the ones had DLT. So that was in the 200 milligram with a 6-day schedule. So 2 patients had DLT. That means they go out of the trial during first cycle, and therefore they're not evaluable for the first CT scan. So that's 2. And 1 patient opted to drop out, and we can't force a patient to stay on a trial. And 1 patient moved to a different part of the country and also lost to follow-up.
Okay.
Yeah, so that's a good explanation. It's we're not trying to hide anything.
Yeah. Okay, thank you, Lars. Now we'll bring up John here again to ask some questions for your field. Thank you for the presentation earlier.
Welcome.
What is, according to you, the advantage of the double mode of action for SCO-101?
Yeah, I think I hope it was clear from the presentation. I'll be happy to, of course, try to explain very simply again. You have an atoxic molecule. It's given as a tablet, so it's easy to take. and you can reduce the amount of chemotherapy that the patient receives, and you can hopefully increase the amount of chemotherapy that the cancer cells exposed to. So I think that is it's a win-win situation, I would say.
Yes, and could there be any drawbacks for this double mode of action?
Yes, potentially there could. I mean, when we started the trial in the first patients, it has never been combined with chemotherapy in patients before. So of course we were very excited and anxious if there would be any, unwanted toxicities or anything. But as Lars very clearly explained here, more than 150 patients has received SCO-101 now, and the toxicities are minor and at least not worse than what you see when you just give chemotherapy. So we have been lucky to not see any unwanted toxicity.
Does it exist, or do you know of any other cancer drugs with this double mode of action?
No, to our knowledge, there is no drugs with this unique mechanism of action, and we had several people to look at that quite detailed, and we have not been able to come across any drugs with this dual mechanism of action. So we think it's unique.
Okay, but please, could you tell us some more about the data that you have obtained in gastric cancer?
Yeah, of course. Yeah, that was a very interesting study, I think, because it was based on the clinical finding that irinotecan is used in gastric cancer. It's also used in other cancer indications, obviously. But the good thing about gastric cancer is that there's also scientific literature demonstrating that ABCD2 is overexpressed in some of these gastric cancer cells, and that this expression has been correlated to the overall survival of the gastric cancer patients. So in that sense, it was kind of a copy from the colorectal cancer to the gastric cancer, and the preclinical data showed quite clearly that when you have high levels of ABCD2, you get this combination effect. When you have low to no levels of ABCD2, you don't see this combination effect. So we think we have demonstrated that in gastric cancer cells preclinically, you get a strong combination effect of SCO-101 and the chemotherapy.
Thank you. And finally, could you tell us some more about the MOA of SCO-201 and the data you have on cancer?
Yeah, we have quite extensive preclinical data on SCO-201, and it targets the ABCD2 efflux pumps really strongly, and it also targets the UGT1A1 a little bit less strongly than SCO-101. So we think that this could probably be a very interesting molecule to test further in cancer. Yeah.
Okay, thank you. And if we could get Monsieur Francois Martelet back up again. Thank you so much for the presentation and all the answers. So I, of course, get a lot of answers of if you have any conversation with big pharma and partnering deals and so on. What can you tell us?
Of course, I mean, we are always in conversation with potential partners, and that's the name of the game, as you know, in biotechnology. So, but I cannot comment any further than that, obviously.
Okay, and before we sum it up here, what would you say, why should investors turn to your company, to Scandion, to put their money?
Well, first of all, you know, as people were saying before, you know, my colleagues, you know, there is an innovative drug, SCO-101. We do have clinical data. We have preclinical data. We do have clinical data that are very much supportive in colorectal cancer and in pancreatic as well. Then we have a very robust IP situation, which is very important. And then we have been going through the necessary steps from a manufacturing standpoint where we are in the phase IIa. Now, moving forward, there is, as Lars was saying, you know, we are moving very quickly towards a randomized clinical trial. And obviously, this is the time to stay tuned, you know, it's not the time to leave the boat, you know.
Okay, so thank you so much, and we all wish you the best of success, especially for those patients with these cancer forms that you have.
Thank you very much.
And the stockholders will be rewarded with that.
And thank you for their patience as well.
Yeah.
Thank you.
Thank you. And thank you everyone that has been watching the broadcast.